Okay, so this is chapter 13, 15 if you have the 8th edition, about the cytoskeleton. And this is a very fundamental chapter. We will be coming back and talking about microtubules and microfilaments in the next couple chapters, especially as we talk about vesicle movement and cell signaling, muscle contraction. So the objective of this chapter is for you to learn some of the basic characteristics of these 3 different classes of cytoskeleton: the microtubules, the microfilaments, and the intermediate filaments. And you have a nice figure here, table here, that gives you a summary, and I've also posted a chart for you to fill out that will help you and be a good reference when you're studying for the quiz or taking the quiz. All right, so let's talk about microtubules. It took me a while to figure out this image, and so what I want you to really understand is that microtubules are made up of alpha and beta tubulin. And you will see on the quizzes and tests, they limit my font choices, and so a lot of times I have to write out alpha and I have to write out beta. It won't give me the symbol fonts. So alpha and beta tubulins come together to form this heterodimer. And hetero means different, and dimer means 2. So it's made up of 2 different proteins. And what's interesting about tubulins, as they come together, is it gives the microtubule polarity. You might remember the word polarity from general biology when we talked about water. Remember water being a polar molecule, right? So polarity means that there is a difference in the end. So they're not interchangeable. Okay. This doesn't have to do with bonds like water, so I'll erase that not to confuse you. But we will be using that term polarity. And so then these heterodimers, beta and alpha, start coming together. Beta, alpha, and they start to form this long protofilament. Okay, and that is right here, protofilament. And then what happens is 13 of those protofilaments start lining up, and they make this hollow tube. So the microtubule is a thick from one, we call them walls, edge of the wall to the other edge of the wall is 25 nanometers. Always make sure you watch your units when you're talking about characteristics, and I could throw in maybe millimeters or centimeters, and so nanometers. These are skinny little things in our cells, and these are the thickest of the 3 cytoskeleton components. So this next slide just shows the microtubules. So when we talk about a microtubule, we're talking about this whole big hollow structure, and you can see them here in an axon, and then sometimes the microtubules actually come together in doublets or triplets, and these are typical of what we call axonemal microtubules, and you will find these at the basal body and in cilia and flagella. Okay. And if you look really closely, you will have one complete 13 protofilament microtubule, and then the other one is not quite complete because they are sharing these protofilaments. So microtubules and the whole cytoskeleton in general is called dynamic. And dynamic means it's ever changing. So it's not like our skeleton, where once we grow, it pretty much stays the same. Yes, we do shrink as we get older, but that's not because our skeleton is shrinking. It's because all the stuff in between our bones are getting compacted. So in your cell grows and shrinks and moves and things like that. So the microtubules have to grow and shrink and be dynamic. The opposite of dynamic is static. Okay? So dynamic means ever-changing. And for microtubules, they can grow at both ends. Oops. But they grow faster at the plus end. So we're going to talk in a minute. Usually the minus end is associated with a microtubule organizing center, MTOC. And so here the dimers are added on and the microtubule continues to grow. But I want you to see that you can lose and you can grow at both ends. And this is just showing an example. So here's a basal body, and this is a kind of this is an example of a MTOC, microtubule organizing center, and the plus ends are growing much faster than the minus ends. Another cool thing that you can observe with microtubules and actually microfilaments is something called treadmilling. So this really illustrates how dynamic this is. So what we're seeing here is that heterodimers are coming off of the minus end and they're being added to the plus end. And so we could use a technique called speckle microscopy, where you can actually inject some fluorescently labeled heterodimers into a live cell, and they will accumulate, right? And so they'll be labeled, in this case they're blue, and as new ones are added, this blue one, and as the minus end is lost, this blue one appears to be moving down the microtubule, right? So what's really happening is this end is getting shorter and this end is growing. But if you can imagine, if you've ever been on a treadmill and that track, I don't know what you call that thing that you walk on, right, goes below your feet like this, right, continuously. So treadmilling is a way we can look at that dynamic nature of microtubules. Another way we can look at microtubules is using drugs. So using chemicals that affect microtubule polymerization. So the ones that are on your chart to know, be familiar with for your quizzes and exams. Colchicine is a drug that is used quite often, especially when we were first studying cell division. So colchicine will bind the beta tubulin, so part of that heterodimer, and stop it from being assembled, right? So your microtubule here no longer can grow. Nacodazole, and I could be pronouncing these incorrectly, does the same thing, inhibits polymerization. Okay, so if you were putting this into a cell, you would see a stop of growth. Taxol here is actually used as an anti-cancer agent, and it does something, it stabilizes the microtubules, right? So this is preventing the microtubules from getting severed, from breaking down, from depolymerizing. So here these microtubules would stop growing, but they could still start breaking down, right? Oops, that's not going to work. Okay. Here it stabilizes. So why would we stabilize microtubules to treat cancer? The hallmark of cancer is uncontrolled cell division. And we're going to talk about cancer at the end of this semester. And so what's happening in cancer cells is they're dividing and dividing and dividing and dividing and dividing and dividing and dividing. Okay, they're not stopping through cell cycle like cells should. Oops, move that guy over here. So if you treat these rapidly dividing cells with Taxol, they stop dividing because they're stuck. The microtubules are stable that are trying to separate the chromosomes, and they can't complete cell division. And one of the hallmarks of cancer cells versus normal cells is that they are more rapidly dividing. And so we use chemotherapy drugs that target cell division. Unfortunately, we also have other cells that are rapidly dividing, such as the cells that line your stomach. So people that are getting chemotherapy often get nauseous. The cells that produce your hair, around your hair follicles. So these drugs also stop those cells from working, kill those cells, and so people lose their hair when they go through chemotherapy. But it doesn't affect the majority of our cells as much because the majority of our cells are not continuously dividing, at least not at the rate of cancer cells. Something I'm going to ask you to do on your homework is draw a chart like this. Okay, and so what this chart, and we're going to not worry about the minus end. It gets too complicated. We're just going to worry about the plus end of these cytoskeleton elements. And so what this chart is showing you is time and size, length change, right? So we start out and you've got a whole bunch of GT-tubulin, so the cells continuously increase in size. And then something happens and they start to depolymerize. This could be due to the addition of any of these first three drugs here. Because remember, and I think, let me peek at my slides, we'll talk about this in a minute. Remember, if there's not new tubulin to be put on the plus end, the minus end can keep being lost, and the plus end can also be lost when there's not this GTP cap, which we're going to talk about. So when you add a drug like this, okay, the length of your microtubules decreases because the plus end is not being added to, and it actually is starting to shrink. Okay. Every time you see these little peaks, this is rescue or resuming growth or polymerization. So if you added something like Taxol, right, you started to have free GTP tubulin and it was working, and you added Taxol, you might expect the length to just level out, right? Because it's stabilizing those microtubules. They're not growing and they're not breaking down. So you will be drawing something like this, but with microfilaments instead. Okay, the GTP cap is a characteristic of microtubule growth. Okay, so this is growth, and remember that the alpha and beta tubulins can bind GTP. Okay. And what's really interesting is if you read the textbook, it says GTP is not required, and it doesn't have to be hydrolyzable, which means to break down to GDP. But in the cell, in vivo, we have alpha beta GTP. And so as those dimers are being added, you get this nice cap and you grow. And then here where it's orange, what's really happening is all of these have GDP. So remember, GTP can be hydrolyzed to GDP and inorganic phosphate, just like ATP. And then they're not going to be added as rapidly as the GTP tubulin. So when you have lots of GTP tubulin, you get growth. When you have low GTP tubulin, okay, you actually lose this protective cap, and the plus end will start to break down. And when this happens rapidly, we call that catastrophe or shrinkage. And again, we can manipulate conditions in the cell to study this. Okay, so you know a little bit about the dynamic nature of microtubules. Let's talk about their work in the cells. So centrosomes and MTOCs, right, are the origins or the initiation sites for microtubule growth. And you will see that the minus end is at the MTOC and the plus end grows away. And this could be an example of cilia with its basal body. These microtubules are probably important in vesicle trafficking. Here's your axon, neuronal cell that we'll talk about in a few chapters. And here are your spindle fibers for cell division. Okay, so if you can see here in the middle are your blue little chromosomes, and hopefully you remember that the spindle fibers attach and they pull and separate the chromosomes for cell division. There's one more type of tubulin in the cell, and that's called gamma tubulin, and that is found at the centrioles. Okay, so let's make sure we understand what a centrosome and a centriole are. So the centrosome is really a region. It is made up of this matrix, so this green fuzz around it, and then the centrioles, which are those doublet, and triplet microtubules. And they are the initiating sites, and here is our gamma tubulin, where the microtubules grow from. So if you look at here, those microtubules are growing out, and at the bottom here is what's shown right here is gamma tubulin and the accessory proteins. Okay, so this is where microtubule nucleation starts. So nucleation means the start of it. I think your book talks about it as these little seeds, right? So you can think of this as the growing center. Microtubules don't work alone, so it's not just a bunch of long protein tubules in your cell. They can be interconnected. Okay. So this is showing two microtubule associated proteins, MAPs. Here's MAP2, and here's a protein called tau. And what I want you to see from this image is that MAP2 is a much longer protein. And so the bundles of microtubules that MAP2 connect are going to be more loosely connected than the bundles of microtubules that tau connects, because tau is a much shorter protein. So microtubules can form bundles. Microtubules can associate with tip proteins. So now this is an extra protein, so it's not just the GTP cap, but it's actually a protein that binds to the positive end of the microtubule, and it decreases, not right, it decreases depolymerization. So it decreases catastrophe. And this has a little bit more information than you need, but what I want you to see is that as you lose the EB1A tip protein, or not EB1A, sorry, EB1, as you lose the EB1 tip protein, now the cap becomes unstable, and this is showing, I think they're little kinesin molecules, pulling off the heterodimers of GDP tubulin, right? So remember, the GTP cap is formed when there's lots of GTP tubulin. We can also have proteins associated with the tip to stabilize it, so even when GTP tubulin is low, you could still have a stable plus end. Tau is a super interesting protein. It's associated with multiple neurodegenerative diseases, such as Alzheimer's and dementia. And what's happening is, here's another diagram showing tau in the green associated with a microtubule. And as tau gets hyperphosphorylated, so this means this is a protein modification adding a phosphate group. So when you add a whole bunch of phosphate groups to tau, it stops interacting with the microtubules. The microtubule becomes unstable and depolymerizes, and then the tau actually aggregates, which means it clumps up, and you get these neuronal tangles, right? So that doesn't look good. If this is supposed to be a healthy neuron reaching out, making connections, you don't want it tangled up. And there's a lot of research into why this happens. Why does, what I said, tau go bad? There are other proteins that can affect or destabilize the microtubules. So catenin is one that severs microtubules. So just like it looks like, it's a knife and it's going to chop it, right? So now you've got a plus end and a minus end and a new plus end and a minus end. And depending on what that microtubule function is, maybe that's good because it's going to network. Maybe it's bad because you've just broken your nice microtubule. Catastrophins, these are on your chapter objectives. Catastrophins would be something that's working right here. They promote catastrophe. They promote destabilization. Another protein in the cell called Stathmin op18. Stathmin op18 actually binds to these heterodimers. Okay, so it's going to prevent polymerization. It's going to prevent assembly because it's binding up those heterodimers so that they can't join and elongate a microtubule. All right, so the last couple of slides I want to talk about is just helping you understand some of the functions of microtubules. Again, we're going to talk about these more in the upcoming chapters, but microtubules are important for cell movement. And I want you to see on this cell, all this orange, yellow is actin. Okay, so we've got microtubules and microfilaments working together. But microtubules can grow and help a cell move. So a migrating cell may be moving to communicate with another cell, may be moving to a new location. And we're going to talk about this with respect to cancer, because if you've heard of metastasis, that has to do with cells moving to a different location in the body. Microtubules are also important for cell adhesion. So they help cells stick together, and they help cells stick to other tissues, other substrates in our body. And they're important in cell communication and signaling. So here's two cells saying, hey, I'm here. Contact inhibition, something that cancer cells lose. Says, you know, I'm here. Go find your own spot. And so this cell now moves away. Microtubules are important. Again, this is an example of cell communication. We will talk about neurotransmitters. And what's happening in this cell is, if you can kind of see it in blue, which I will now make red. Oh, that's ugly. Let's see if I can do a pointer. Alright, so I'll highlight it now. You still can't see it. Alright, anyways, the blue are the microtubules, and what we're going to talk about next chapter is the microtubules actually moving all these little transport vesicles. And for neurons, what's important is that they move the transport vesicles to the end of the axon and release those neurotransmitter chemicals. Microtubules are important, like I said, for cell division. So when we talk about cell division, we'll talk about polar microtubules and kinetochore microtubules and astral microtubules. And they're all important in helping that cell divide into two. Again, these are targets for cancer drugs. This is just another diagram that we will come back to again in next chapter about microtubules being important for vesicle movement. So we've talked about anterograde and retrograde transport or movement of vesicles. Okay. So we're going to learn that there's actually these cool little motor proteins that carry these vesicles, and they carry them along the microtubules. So I like to think of the microtubules as the railroad tracks of the cell, and so it gives these guys little destinations. Okay, you go to the plasma membrane, now you go back maybe to the Golgi or the ER. And last, another function of microtubules in cell movement is moving things around the outside of the cell. Okay. So, oh, and I have been spelling it wrong, cilia with one L. I knew that, and then I don't know. Okay, so some cells, like in your lungs, not lungs, the trachea, have these cilia projections that help sweep the air and microbes. Air into your lungs helps sweep microbes out of your lungs. So cilia are these long microtubule projections. They come from a basal body, and they have, you can see, these unique duplet groups of two microtubules together. Flagella, you know, from a sperm, right? So it helps the cell move through its environment. Cilia, really super important. These little guys get damaged by cigarette smoke, air pollution, and there are actually some pathogens that will damage the cilia so that they can attach. So pertussis, whooping cough, is caused by a bacteria that actually has a toxin that damages these cilia so that you can't cough it out and sweep those microorganisms away. Okay, I'm going to stop there. I'm going to get something to drink so I don't choke, and I will come back with microfilaments and intermediate filaments.